We'll get started today. So today is the last tutorial for the year. I'm pretty sad about that. So today we'll go through exercise, diabetes, and obesity. So all of these ones we're putting everything that we've learned together. So everything we learned, like glycolysis, citric acid cycle, beta oxidation, all of that comes into it. So first of all, we'll talk about anaerobic exercise. So anaerobic is high intensity exercise for short periods of time, where most of the energy is generated from anaerobic respiration. So this is the production of ATP without using oxygen. And so that's the process of glycolysis. I'll just ask him to turn the volume down a bit. Hey guys, would you just mind turning down the volume a little bit?
Yeah, sure, sorry.
Yeah, thank you. Sorry about that. So this occurs in type 2 muscle fibers, and there will still be a little bit of aerobic respiration going on that contributes to energy. So the energy is going to come from glycogen and phosphor creatine. The pyruvate that's produced from glycolysis is turned into lactate or lactic acid in order to regenerate NAD+, which is critical to allow glycolysis to continue. Okay. The lactic acid is transported in the blood to the liver, where it turns back into pyruvate, and the pyruvate will be turned back into glucose through gluconeogenesis, and then that's going to be released into the blood again. And so how many ATP are produced per glucose from glycolysis? So, yeah, 2. So that's not very efficient. So you're only getting 2 ATP per glucose if you only do glycolysis, because that pyruvate molecule still contains a lot of energy. So we have the blood, and so the blood's going to contain glucose, and that's going to be taken up by this muscle cell, or the myocyte, and in this case it's a type 2 muscle cell. So once it gets in, it's going to be broken down through glycolysis, and through glycolysis we're going to take ADP and we're going to turn that into ATP. And during this process, we also need NAD+. So we need an NAD+, and that's going to be reduced and form NADH. So remember that glycolysis cannot occur if you don't have enough NAD+. So if you only did this, we'd run out of NAD+. So we have to turn NADH back into NAD+. So this is, at the end of glycolysis, you end up with pyruvate, and that's going to be turned into lactate, and that process there will enable you to regenerate NAD+. So inside this myocyte, we're going to have a store of glucose, but we can't store it as glucose. What would we store it as? Yep, glycogen. So we have glycogen being broken down into glucose inside the muscle cell, and that acts as a store of glucose. Because when you need to generate a lot of energy, you need a lot of glucose, and it's going to be hard to get that all from the blood. Okay? And there's also another store of energy inside the cell, so that's phosphor creatine. And so there's a phosphate on the creatine that can be donated to the ADP. So the phosphocreatine will lose its phosphate and give it to ADP, and so you'll get ATP and creatine from that reaction. So type 2 muscle fibers, they are fast twitch. So fast twitch, so that means that they can generate a lot of energy. And they are lighter in color, so they're white, because they don't have as much myoglobin. So if you look at like chicken, for example, chicken meat, like you've got some parts of it is white, whereas some parts of it is more brown. So the lighter parts is going to be type 2 fibers, and the darker parts are going to be type 1 fibers, and that's because of the difference in the content of myoglobin. Okay, so they don't need myoglobin, these fibers, because they don't use much oxygen. Remember, they're doing anaerobic respiration. And they're going to have low numbers of mitochondria because they don't do much beta oxidation, citric acid cycle, and the electron transport chain. So remember, all of those occur in the mitochondria. So if you're not doing that, you don't need to have those organelles, just taking up space. And these cells will also have a lot of glycogen. And the enzymes involved in glycolysis, so the glycolytic enzymes, will be high in these cells. Okay, so we'll move on to the next page. So creatine, skeletal muscle stores phosphocreatine, which can donate its phosphate to ADP to regenerate ATP. So this is a very fast reaction, so you can regenerate ATP very quickly. It's normally used up in the first 5 to 10 seconds of very intense activity. So that's depending on your level of creatine stored. So if you take creatine supplements, you can increase the amount of creatine or creatine phosphate that's stored in your muscles. So that might give you a little bit more. You might be able to generate intense energy for a little bit longer, but it's not really going to help you for running a marathon because it's only a few seconds. So it's built up using ATP when you're resting. Okay, so when you're resting, you're going to build up your levels of phosphocreatine so that it can be ready for when you're exercising. And creatine can be created in the liver and kidneys from amino acids, and it can also be absorbed from the diet. Okay, so red meat contains a lot of phosphocreatine. So there's just a diagram there, and the enzyme that catalyzes the formation of creatine phosphate is creatine kinase. Okay, so kinase, remember, means you're phosphorylating a protein, and that's using the energy of ATP. So then we need to know about adenylate kinase. And so if there's lots of ADP due to ATP breakdown, the ADP can be further broken down to produce more ATP, and that's catalyzed by the adenylate kinase, which is also known as myokinase. Okay. So then we have a question at the bottom. Phosphocreatine is found in muscle. Which statement regarding phosphocreatine is incorrect? So A, the amount of phosphocreatine in muscle is approximately sufficient to run a 100-meter sprint. So is this true or false? Yep, so it's true. So remember we said it can generate up to 5 to 10 seconds. B, creatine kinase converts phosphocreatine to creatine and ADP to ATP. Is true or false? Yep. And then dietary supplements providing creatine enhance the performance of marathon runners. Is that true or false? Yep, false. So it would provide barely any benefit, maybe just for the first few seconds. If you're a weightlifter or a bodybuilder, it might help you, but not if you're an endurance runner. So D, creatine is obtained from the diet or synthesized in the body, so that's true. It can be made in the liver and absorbed from the diet. So now we'll talk about aerobic exercise. So this is low intensity for long periods of time. Low intensity for long periods of time. So most of the energy is generated from aerobic respiration. And this is the production of ATP using oxygen. So the pathways, the metabolic pathways involved are beta oxidation, the citric acid cycle, and the electron transport chain. So there will be a little bit of anaerobic respiration contributing energy as well, especially at the start of exercise. And the energy is going to come mostly from fatty acids. Okay. So if you're on the treadmill for an hour, usually you'll use up all your glycogen in your muscles by about the first 20 minutes or so. And then you'll gradually, so it takes time so remember when you start exercising, you're going to release adrenaline, and adrenaline is going to stimulate hormone-sensitive lipase in your fat cells, but all of that takes time, so it takes about 20 to 30 minutes for it to kick in. So I don't know if you've experienced this, but if you run or do any form of exercise, you might be able to go hard for maybe 10 to 15 minutes, and then you might kind of feel tired again, and then after about 20 minutes you start to feel more energy. So that's when your fatty acids are kind of like kicking in, because all of your citric acid cycle, your beta oxidation, all of that kind of takes a little bit of time to kick in. So that's kind of where the term the second wind comes from. Fatty acids are broken down via beta oxidation to produce acetyl-CoA. So acetyl-CoA enters the citric acid cycle, which produces 1 GTP. And what else do you get out of the citric acid cycle? Yep, and yep, that's right. So you get 1 FADH2, and you get 3 NADH. So NADH and FADH2, remember they're carrying the high energy electrons, and they get oxidized. So oxidized means they get the electrons taken off them at the electron transport chain, and as a result of that, ATP gets produced. So oxygen is necessary for the electron transport chain to work, and this is why it's called aerobic. Okay. So you can see in the diagram there that, again, we've got a bloodstream, and this time we're going to be using fatty acids. So the fatty acids are going to diffuse into the cell, and we also need oxygen this time. So there's going to be oxygen in the blood that needs to diffuse into the cell. Okay, and so we've got the fatty acids coming in, and they are going to go into the mitochondrion. So remember, the beta oxidation occurs in the mitochondria. So what's the product of beta oxidation? Yep, acetyl-CoA. And then that's going to go through the citric acid cycle. It's going to go through the electron transport chain, and that will produce ATP. Okay. So this oxygen will take the electrons, so it's the terminal electron acceptor, right? And what does it form? Water. Yeah, water, so H2O. It's going to take 2 electrons and 2 protons and become H2O. So we need a lot of oxygen for this to happen. So if you can remember to the start of the semester, we learned about myoglobin inside the muscle cell. So this is where it's coming in. These muscle cells contain myoglobin, and these store a lot of oxygen. So these can contribute to the supply of oxygen. Okay. Right, so type 1 fibers are slow twitch, so they're more for endurance. You're not going to do any, if you're doing an explosive workout, you're not going to be using your type 1 fibers. These are slow twitch, and they're darker in color. They're red because they have a high concentration of myoglobin. So remember, myoglobin and hemoglobin is what gives the blood and muscles its red color. Okay. So the myoglobin stores oxygen, and this is what allows aerobic respiration to occur. And they're going to have lots of mitochondria because we need mitochondria for beta oxidation, the citric acid cycle, and the electron transport chain. And so, next page, which of the following properties is not a characteristic of skeletal muscle of athletes that have undergone regular aerobic exercise training? So this is aerobic training. So A, would they have a high myoglobin content? Yep, they would. So remember, they're doing aerobic exercise. So their cells are going to adapt, and you're going to get an increase in your type 1 muscle fibers, which are slow twitch, and they're going to, if you're doing aerobic, you need a lot of oxygen. So B, would you have a high capillary density? Yep. So remember, capillaries, these are the capillaries, and you're getting oxygen from the capillaries. So the more capillaries you have, the more oxygen that you can receive, and the more waste products they can take away as well. So C, would you have an increased number and size of mitochondria? Yep, you would. And then D, would you have a high glycolytic enzyme activity? No. So that would be your type 2 muscle fibers. So D is incorrect. So insulin, normally blood glucose is kept at a constant 5 millimoles a liter. Okay, so no matter what you do, no matter whether you exercise or whether you eat, if you eat a lot of sugar, it can go up to like 7 or 8 millimoles a liter, but it'll quickly come back down to 5 millimoles per liter. And insulin is critical to achieve this. And if you actually go below 4 millimoles a liter, you'll notice it like you'll notice it pretty severely, because if you've ever exercised quite hard after you haven't been exercising a lot, you can feel nauseous and really trembly. So if you've experienced that, you've gone hypoglycemic. So when you exercise, your muscles are taking up all the glucose from your blood, and that's going to lower the blood glucose. And so your brain senses that, and it produces a whole heap of adrenaline to trigger glycogenolysis, to trigger fatty acid oxidation and lipolysis. And adrenaline causes you to tremble, and it also causes you to feel nauseous and feel like you're going to vomit. Okay. So insulin has a wide range of effects in the body. Its main function is to lower blood glucose levels. That's why it's released when so it's released by beta cells of the pancreas in response to elevated blood glucose levels. So you can say elevated or high or increased. Insulin is required for skeletal muscle and adipose cells to absorb glucose. Insulin binds to the insulin receptor. So it's a hormone, so it's going to bind to a receptor. And this tells the cell to put glucose transporters on the cell surface. So it's important to remember that normally on an insulin-dependent cell, so like muscle cell and fat cells, normally there's no glucose transporters on the cell membrane. They're all inside the cell. And then when insulin binds to the insulin receptor, that's going to tell the cell, put the glucose transporters on the outside, and then once they're on the cell membrane, they can start transporting glucose into the cell. Okay. And so insulin, so one of the effects is to tell the cell to put glucose transporters on the cell surface, but it does a whole lot of other things as well. So it inhibits hormone-sensitive lipase. We've gone over this a number of times, but it's important to remember that. And that's the lipase in adipose cells, and so it inhibits lipolysis. So lipo for fat and lysis for breakdown. This prevents the release of fatty acids into the bloodstream. So why would you want to prevent the release of fatty acids into the bloodstream? Yeah, exactly. So you've got a lot of glucose in your blood already, so you want the cells to use glucose for energy, not fatty acids. So you don't want to be breaking down more fat. And it's also going to inhibit gluconeogenesis. Remember, you've got enough glucose, you don't want to be making any new glucose. And it's also going to inhibit glycogenolysis. So so glycogenolysis, what is that? Yeah, so that's breaking down glycogen. Again, you have enough glucose, so you don't need to break down glycogen. Insulin is going to stimulate lipogenesis. Lipogenesis, so that's making fat. You've just eaten, you've got a lot of energy, you want to store that for later on. And that stimulates glycogenesis, or the formation of glycogen as well. And it also stimulates protein synthesis, or muscle growth. So it also stimulates your muscle cells to take up amino acids from the blood, because if you've just eaten and you have a lot of sugar in your blood, it's likely that you're going to have a lot of amino acids in your blood as well. So it's encouraging growth while you have the energy available. So then, if you look at that diagram, on the left it has no insulin. So insulin has not bound to the insulin receptor. So you can see that inside that cell is the vesicle that contains the glucose, the GLUT4 transporter. And so there's no GLUT4 on the plasma membrane, which means that the glucose cannot go into the cell. And so this is a protective mechanism. So remember that if your brain always requires insulin. So your brain doesn't need glucose. It has your brain has glucose transporters that are always present, no matter whether you have insulin or not. Okay, because the brain can only use glucose or ketone bodies for energy. So when you have low blood sugar and you need to keep that glucose in the blood for the brain, so you don't want your muscles to take up all your glucose, so that's why you require insulin on your muscles to take up glucose. So when insulin is present, it's going to bind to that receptor, and that's going to, the fusion of the vesicle with the membrane allows the GLUT4 to be expressed on the membrane, and that's going to allow the uptake of glucose. So we have a disease called diabetes, and there's two types of diabetes mellitus. There's type 1, which is where there's no insulin production. And this is due to the destruction of beta cells in the pancreas. So remember, you have alpha cells and beta cells in the pancreas. So beta cells create insulin, alpha cells create glucagon. Okay? And that's due to an autoimmune response. So your immune system goes a bit wonky and attacks your own pancreas for whatever reason. And this means that you don't produce any insulin. And remember that insulin is very important in keeping your blood glucose levels regulated. So they're going to be very high. So they can be up to 30 millimoles a liter. And if they are up at 30 millimoles a liter, that's very bad. And this usually happens at a young age. So the usual age of onset is about 10 to 15 years old. You don't need to remember like the exact numbers, but just it's a young disease. It's quite rare. If you've gotten to the age of like 30 or 40, you're probably never going to get type 1 diabetes. Okay, so if you've made it this far, it's very low chance that you'll get type 1 diabetes. I'm sure you all know at least a few people with type 1 diabetes. I know quite a few people. So it's relatively common. Okay, so it can be treated with insulin injections. But insulin is a very powerful hormone. If you give too much, this can result in hypoglycemia or low blood glucose, which can be fatal, remember? So if you lower your blood glucose too much, then there won't be enough glucose for your brain. And then if the brain starts to die, then yeah, it's not a good thing. So now we'll talk about type 2 diabetes, which is all about insulin resistance and decreased insulin production. So type 1 and type 2 are very different in their onset. So the cells become resistant to insulin, so they stop responding to insulin. During the development of diabetes, insulin levels are going to be increased initially in order to overcome this resistance. Okay, so imagine that you've just had dinner, right? So you're really full, and then someone comes along, so this is insulin, comes along and tells you to eat more. You're not going to eat more because you're already full, right? But if insulin is increased and it really forces you to eat more, then you'll eat more. So insulin levels are increased in order to overcome this resistance. Continued high production of insulin leads to the beta cells burning out. And then once they start to burn out, the insulin levels will start to decrease. And when they start to decrease, this is the start of diabetes. And the insulin levels will continue to decrease over time if not treated. So it's usually due to obesity and an inactive lifestyle. So if you're eating too much and you're not exercising, then your cells will have all this energy that they're not using, so they don't want to take in any more glucose, so that's why they stop responding to insulin. Okay, so that's why it's important to not eat too much and exercise a lot. Okay, and type 2 diabetes occurs later on in life, usually after 40 years of age. Okay, so in the exam, if you have a young person and they have elevated blood glucose, it's most likely going to be type 1 diabetes. Whereas if you have an older person who's overweight and doesn't exercise, it's going to be type 2. Okay. So the treatment is to improve insulin sensitivity by losing weight, exercising more, and medications. So the good thing about type 2 diabetes is that if you change your lifestyle, then you can actually cure yourself of diabetes. If you've left it too late, then you might not be able to completely cure yourself, but you can definitely, type 2 diabetes is one of those diseases that you can influence a lot just by changing your lifestyle, okay? Because it is a lifestyle disease, and this is a huge problem in New Zealand and all over the world. So type 2 diabetes is probably going to take up, within we had a lecture on this, and it's probably going to take up 60% of our health budget within the next 50 years. That's how big of a problem it is, because type 2 diabetes affects your whole body. It's a very nasty disease, but it's completely preventable. There is a genetic component, but it's mostly preventable. So yeah, you guys should keep that in mind. Okay, so hyperglycemia or high blood glucose. So if you don't treat the diabetes, if you just let it go, so the normal range is around 5 millimoles, but diabetics will be far above this value. And it can lead to vascular disease. The high concentration of glucose damages the blood vessels and can lead to decreased blood flow to the limbs and blindness due to damage to blood vessels in the eye. So if you've had golden syrup or maple syrup, it's sticky, right? So if your blood sugar levels are really high, your blood's also going to be quite sticky as well, and it's not going to flow as well. It's going to damage the blood vessels, the walls. Increased lipolysis. So insulin normally inhibits hormone-sensitive lipase. So if you don't have insulin, or your cells aren't listening to insulin, the hormone-sensitive lipase is going to be more active, and this is going to result in lots of fatty acids in the blood. And that's going to make things worse, because you've already got a lot of glucose in your blood, and then you're releasing more fatty acids. So that's not going to be a good thing. Ketogenesis, you have increased levels of acetyl-CoA because you've got increased fatty acids and they're being broken down through beta oxidation. And the buildup of acetyl-CoA causes the synthesis of ketone bodies. And the production of acetone from ketone bodies gives the breath a fruity smell. But that's pretty end stage, so that's towards the end. If you can smell a fruity scent, then that's quite far advanced, and they should be in the hospital. So acidosis, low blood pH, the ketone bodies are acidic, and so they'll lower the pH of the blood, and your pH is very important because if your pH becomes lower, there's more protons in the blood, and they're going to protonate your proteins, and if your proteins get protonated, what happens to them? So they'll change shape, because remember, you've got a lot of these proteins require them to be in the right charge for the right interactions. So if you remove that, if you distort that charge, you're going to change the shape of that protein and it's not going to function properly. So you're also going to have dehydration due to large volume of urine. So if you have excess glucose in your blood, it's going to get peed out, which is going to pull out water with it. And you'll also have muscle wasting. So remember that insulin promotes protein synthesis. So if you don't have insulin, you're going to get muscle breakdown. And if insulin is not present or muscles aren't responsive to insulin, then the muscles will start to break down. So here's an exam question from last year. We've got two 20-year-olds who have drunk some beer. And then at the hospital, they got their blood checked. So they got their blood alcohol and their blood glucose checked. So if you just look at the glucose, subject A has a blood glucose of 5 millimoles per liter. So that's normal, right? But subject B has a blood glucose of 17 millimoles a liter. So that's way above normal. So they measured it again the following day, and it was still very high. So it was still 16 millimoles per liter. So what could account for these blood glucose levels in subject B, and what treatment is likely to be recommended? So you can take a guess.
Type 1 diabetes?
Yep. So it's most likely type 1 diabetes because he's quite young and he's got elevated blood glucose levels, consistently elevated blood glucose levels. Your blood glucose levels can get up to 10 or 11. That's okay. But they should come back down to 5 millimoles per liter if you haven't eaten for a while. Okay. So on the next page, so the treatment for type 1 diabetes, what would you give them? Yep, insulin. So they're going to be insulin injections. Now we're on the next page. Insulin is released from the, where is it released from? Yep, that's right. So they just want the tissue, so it's going to be the pancreas. And that's going to be in response to low or high blood glucose. Yep, high. And the binding of insulin to its receptor in muscle and adipose tissue promotes the movement of a glucose what to the cell membrane? Yep, transporter. A lack of insulin or decreased responsiveness to insulin causes the disease diabetes. Yep. Well, to be specific, it's diabetes mellitus. Okay. Because there's another type of diabetes called diabetes insipidus, which is quite a different disease. But in that disease, you also get a lot of urine being produced. Okay. So diabetes means like a siphon, so water's just being siphoned out of you. And mellitus means sweet. So if you taste the urine, which is what they did back in the day, so back in like the 1600s, because they didn't have like fancy blood tests like we do now, one of the ways to diagnose someone was to taste their urine. And if it was sweet, then that person had diabetes mellitus. So luckily, we don't have to do that anymore. So list 3 metabolic processes that are blocked by insulin. So who can list? Just list one. Gluconeogenesis? Yep. So you don't want to create more glucose when you have enough glucose. What's another one? So we want glycolysis because we want to break down glucose. What would you want to break? What wouldn't you want to break down? Yeah, glycogen. So glycogenolysis is going to be inhibited by insulin. And what else would you not want to break down? Sorry? Yeah, so what's that called? What's the breakdown of lipids called? Yeah, lipolysis. Great, so it's lipolysis, gluconeogenesis, and glycogenolysis. So they're inhibited by insulin. And there's a whole heap of other stuff. So you could also say ketogenesis as well. That's another one that's inhibited. So the production of ketone bodies. So explain why insulin cannot be given orally. So I talked about this back at the start of the semester. Yep. Yeah, so it's a peptide hormone. So it's going to be, it's a chain of amino acids, about 50 amino acids long, and in order for it to work, it has to be intact. But if you put it in the GI tract and the proteases attack it, it's going to get broken down into 1 or 2 amino acids, and then it's absolutely useless. Okay. So then we'll talk about obesity. We have an obesity epidemic all over the world, and over 20% of adults in New Zealand are obese. There's a number of factors. There's the poor diet, lots of fast food, decreased exercise, and genetics. So genetics do play a big part in obesity as well. That's undeniable. But that's in conjunction with the environment. Okay. The BMI is your body mass index, and that indicates whether you're underweight, normal, or overweight for your height. So your normal BMI is 18.5 to 25. And what's the cutoff for the BMI if you're obese? Yep, 30. So if your BMI is over 30, you're classified as obese. And nearly 1/3 of New Zealanders are obese according to this definition. But the BMI can be misleading. So if you took the BMI of all the All Blacks, nearly all of them would be considered obese, but they're just carrying a lot of muscle mass, right? So you wouldn't say that any of the All Blacks are obese. But if you were just wanting to analyze the general population, the BMI is your most convenient and accurate measure. So that's what we use. There's always going to be exceptions, okay? But just keep that in mind. So the BMI isn't your only measure of health, but it's a very easy one to measure. So the complications of obesity, the risk of type 2 diabetes increases dramatically, and you're going to also have an increased risk of heart attacks, and you're going to have an increased risk of hypertension or high blood pressure. So in terms of biochemistry, there's a few things that they want to target or that can help in increasing your metabolic rate. So you've got brown fat, which contains uncoupling proteins. So the brown fat has proton pores in the inner mitochondrial membrane. So this allows the uncoupling of the electron transport chain from the production of ATP. So remember that the electron transport chain, what was the purpose of the electron transport chain? Yep. So the purpose of the electron transport chain is to generate that proton gradient between the intermembrane space and the mitochondrial matrix, and that proton gradient or the proton motive force allows the generation of ATP by allowing the flow of protons through ATP synthase. But if you have, so the second bullet point there, this allows the consumption of energy through beta oxidation of fatty acids, citric acid cycle, and electron transport chain to generate heat without ATP being produced. So these uncoupling proteins are just letting the protons through without producing ATP. So that way you're not producing ATP, so you're not inhibiting the electron transport chain, and you're not inhibiting the citric acid cycle. So you can, so this is a way to elevate your basal metabolic rate. And the researchers are trying to find out if we can activate brown fat. So babies, when they're newborns, have a lot of brown fat, but as you get older, you lose brown fat and it turns into white fat, which doesn't have this ability. So it's similar to the effect of DNP, but DNP is quite dangerous because it can cause you to, it's very easy to overdose, and you can it's quite easy to die from hypothermia. You'll literally cook yourself from the inside out. So then we've got leptin. So leptin is produced by the adipose cells. So adipose is another name for fat when they are full of triglycerides. The leptin binds the leptin receptors and causes a cascade of reactions, or in other words, a signaling pathway that signals to the brain that we are full. So the precise location in the brain is the hypothalamus. Okay, so you should write that down. It's in the hypothalamus. So the leptin receptors are located in the hypothalamus. And remember, the hypothalamus is kind of like the control center for your whole body. It detects osmolarity. It detects your satiety and a lot of other things. So it tells the brain that we have enough energy, so we should stop eating and increase activity. And this is a mechanism that prevents excessive energy intake. So if you have problems with leptin production, it can cause obesity because your brain's never getting the signal that it's full. So children with no leptin production will be severely obese, as they will never feel satiated or full. And they can be treated with leptin injections. But being unable to produce leptin or leptin deficiency is actually quite rare. So what this means is that most obese people don't have a problem with producing leptin, and they actually have lots of leptin. And it's actually quite similar to insulin. So they have so much leptin that the body kind of gets resistant to the effects of leptin. And there can also be problems with the leptin signaling pathway, which means that even though there's enough leptin being produced, the signal isn't being generated in the cell that we are full. So this can also lead to obesity. So on the last page, we've got a few questions. So which statement regarding uncoupling protein is incorrect? So A, uncoupling protein was discovered in a specialized thermogenic tissue known as brown fat. Yep, so true. B, uncoupling protein allows the movement of protons across the inner mitochondrial membrane and therefore collapses the proton motive force. Yep, so that's true. The ATP produced in brown adipose tissue in the presence of uncoupling protein is used to produce heat in hibernating animals and human babies. Yep, that's true. And the presence of uncoupling protein increases metabolic rate via uncoupling ATP synthesis from fatty acid oxidation. Is this true or false? So this is false. It could be very easy to think that this is true, but remember that it's uncoupling ATP synthesis from the electron transport chain, not fatty acid oxidation. Okay, so it's uncoupling ATP synthesis from the electron transport chain. So leptin is a hormone secreted by adipose tissue that regulates food uptake. Which one of the following statements concerning the availability and action of leptin is incorrect? So A, an obese individual who has a homozygous mutation in the gene encoding leptin can be treated by leptin injections. So is this true or false? Yep, true. So this person has a leptin deficiency. He can't produce leptin, or the leptin that he produces is defective. So if we give him good leptin, then we can help him. B, leptin can be prepared through recombinant technology, allowing leptin to be obtained by expressing the cloned leptin gene in appropriate cells. Is this true or false? Yep, true. So we learned about recombinant technology earlier on in the semester. So make sure you know about that. Make sure you know about the different cell types that you can produce proteins in, because they like to ask about that. An obese individual will be insensitive to leptin injections if there's a mutation in the leptin receptor that prevents the binding of leptin to the receptor. Is this true or false? Yep, true. And D, leptin injections will be beneficial in obese humans who have a mutation in a gene encoding a protein in the leptin signaling pathway. Yep, so it's false. So in this person, the leptin is fine, the receptor is fine, so the leptin can bind to the receptor, but it's the signaling pathway inside the cell that's defective. So giving them more leptin isn't going to help the problem. So next question, leptin is a hormone that when present provides a signal to the brain to decrease food intake. Which statement is incorrect? So A, experiments with obese rats identified a homozygous mutation in the gene for leptin. True or false? Yep, it's true. And B, in humans, leptin is secreted from adipose tissue. Yep, it's true. So when is it secreted from adipose tissue? When the cell is full or empty? Yeah, when it's full. So C, the leptin receptor is in the hypothalamus. Yep, that's true. And D, patients lacking the leptin receptor can be treated by regular injections of leptin. Yep, that's false. Cool. So that's everything. So thank you very much for your attention this year. It's been a pleasure to be able to see all of you every week, and you've been very respectful, and it's just been a real pleasure. So I'll miss you guys, and if you see me around next year or later on in the year, please say hi, because sometimes there's so many of you, and I'm just like, I don't know if I'll like kind of recognize you, but I'm not sure. So, but if you say hi and just say like, oh, I was at Carolyn Freeman, then I'll be really happy to say hello to you. Okay, cool. All right, thanks guys, and good luck for exams.